Welcome back to the Banajwar College video classes. In my last lecture, I have explained about the structure of the muscle and as well as the structure of a single myofiber. And we have seen that in a single myofiber, there are hundreds of parallelly running, very long, from one end to another end of the myofiber, there are many myofibrils, and each myofibril is made up of the contractile elements or contractile proteins. They are also called as myofilaments. And we have seen that there are two types of the contractile protein, myosin and actin. So today we'll first take up the structure of the proteins, that is contractile proteins, and then the sliding filament theory. So that means today we are talking about the mechanism of muscle contraction, how the muscle contracts. So to understand the mechanism of muscle contraction, we should be very well thorough with the structure of the contractile proteins. We know that actin filaments are the thin ones which you find in the I band or isotropic band of a sarcomere, as you have seen in my last lecture. Let us take up one such thin filament, the actin filament, and know about its structure. It's a double helix made up of the two chains which are called as F-actin chains. And these two F-actin chains are made up of the single monomer that is called as G protein. So the G proteins, they join together forming long chain-like structure, and such two chains will make up one molecule of the actin filament. And each of the G protein, that is in the F-actin chain, each G protein has got a myosin binding site. Okay? And these sites are actually covered by another protein called as tropomyosin. You see here long thread like another protein. So tropomyosin will be running along with the F-actin chains, and they will be covering or blocking the active sites of the actin protein. Then another structure you find attached to the tropomyosin molecule, that is called as troponin, made up of three protein molecules there, three protein units, subunits I should say. So that is called as troponin molecule. And these troponins are the important structures which have got the site for binding of the calcium ions. We'll come to know the importance of the troponin molecules when we take up the mechanism of muscle contraction, that is sliding filament theory. Okay? Now just remember the structure. In case of the smooth muscles, there are, in place of the troponin molecules, there are clavulins. So we'll discuss about that later on. Just remember, clavulin molecules are there in the smooth muscles, but in all other muscles we find that troponin molecules are there attached to the tropomyosin thread. Okay, that is about the actin filament structure. Let's take up the structure of myosin filament. Unlike the actin filament is one protein molecule, a double helix, but the myosin filament is a polymer. So many protein molecules, they combine together forming a polymer of proteins. And here a single unit if you take up, a single structure out of this monomer. We can say it as a myromyosin molecule. So these myromyosin molecules, if you look at the myosin structure here, their heads are emerging out here above the surface of the myosin filament. So what these are, let us see. So this is one myromyosin molecule. It has got again two chains helically coiled, and the straight chain, what we see here, this is called as heavy myromyosin molecule, and even the two heads are also taken as heavy chains, and the connecting, a cross-linking chain, what we have here, which I have drawn in blue color, that is called as light myromyosin. It is at this point that this myromyosin molecule can show movements. These heads can bend inward and extend out. So to regulate those movements, facilitate that one, so there is a light myromyosin molecule. If you look at the head of the myromyosin molecule, these are the heads of myromyosin. It has got two spots. One is the site for actin binding. So with this side, we can say it will bind with the actin. And another side, which I have drawn here in green color, this spot is the ATP, adenosine triphosphate molecule, ATP binding site. So these are very important regions in the head of the myromyosin, and head of myromyosin also contains an enzyme called as ATPase. Myromyosin molecules together make up the thick filament. We call it as myosin filament. Now we have seen the structure of the actin and myosin filaments. Now let us take up the sliding filament theory, because these two types of contractile proteins are important to understand how sliding filament theory, or how the muscle contraction is taking place. That is very well explained by a theory called as sliding filament theory. Let's take up that one. Now let us know about the sliding filament theory. This is the famous theory, very well accepted theory for understanding the muscle contraction. As we know that without muscle contraction, we cannot move even a finger of our body. Any part of our body, if you are making it to move, it is by the help of muscle contraction, and that is very well explained by sliding filament theory, which was proposed by in I think 1954. During that it was proposed by two teams of scientists, one from Cambridge University and another from the University of Massachusetts. So Andrew F. Huxley and Rolf Niederturkey and Hugh Huxley and Gene Hansen. So these are the scientists responsible for this theory, explaining this theory. And what is the main basis of this theory? As we have already studied that in a myofibril, okay, so this is the sarcomere, and we have here the actin filaments, let me take it in the same way. Yes, so these are the actin filaments we have seen, and the myosin will be in the center, And we know that these are the Z lines, and the actin filaments continue into the next sarcomere also on the other side of the Z line. These are the myosin, and these are actins. So what actually happening is, when a muscle contracts, there is a sliding of these actin filaments in between the myosin. So they move inward. Now, how it appears, let us see. The same structure now, after the contraction has taken place, how it appears, let us see. Even this side also, the sarcomeres on the either side, they also show same thing. So what we see is the length of sarcomere is this much here. After contraction, we see that it has reduced. So in this way, the whole myofibril is contracting, and all the myofibrils are contracting, so that the complete myofiber contracts, and one muscle fiber doesn't contract independently. All the muscle fibers in a particular muscle get contracted at a time, bringing about the movement. So that is how the muscle contraction is taking place. We see here that the actin and myosin gets totally overlapped. That is by sliding in of the actin filaments. So this can be understood by these diagrams here. Now first of all, we must know how the muscle is activated. Now this is the neuromuscular junction. Neuromuscular junction. And this neuromuscular junction is a junction between the motor neuron and the surface of the muscle cell, or the plasma lemma of the myofiber. And plasma lemma of the myofiber has got some infoldings where the plasma membrane is rich in the channels, ion channels, and when the action potential reaches the axon ending of the motor neuron, what we see is the acetylcholine molecules, which are the acetylcholine is a neurotransmitter chemical. So these are released, and they will bind to the receptors on the sarcolemma of the myofiber, and that's how the action potential is generated inside the sarcolemma, or the plasma membrane of muscle fiber. Positively charged ions increase, so action potentially are studied before also. So that action potential is generated, and that action potential is carried by the T tubules down to the sarcoplasmic reticulum. And this sarcoplasmic reticulum gets activated. This is a T tubule, which are formed by infolding of the plasma membrane only. And these T tubules are located all along the myofiber, usually near the Z lines or near the myosin filaments. So these T tubules will stimulate or carry the action potential up to the sarcoplasmic reticulum. Sarcoplasmic reticulum gets activated and sarcoplasmic reticulum releases calcium ions. Calcium ions play a very important role, and the calcium ion acceptors are the troponin molecules in the striated muscles or the skeletal muscle fibers, right? And in smooth muscle fibers, climidulins are the calcium acceptor. So this calcium goes and binds to the calcium binding subunit of the troponin. So this is troponin, as we know, troponin of the actin molecule. Okay, and when the calcium ion binds to the calcium binding site of the troponin, that troponin structure undergoes conformational change, and because of the binding of the calcium, what happens is the tropomyosin will be pushed. Tropomyosin will be pushed away from the active sites of the G molecule, G protein molecules or G actin subunits in the actin molecule, so that the active sites of the actin molecules are opened. And when the active sites are open, another thing is now happening, that to the ATP binding site, the adenosine triphosphate, the currency of the energy in any cell if you take up, here also we have the ATP molecules being generated and stored in the cytoplasm of the myofiber, or in the sarcoplasm we find ATP. ATPs are generated by breaking down the glucose, right? In the presence of oxygen, the glucose is broken down to generate large number of ATP molecules. So one ATP molecule binds to the ATP binding site of the myromyosin head. So we know that this is myromyosin head. Okay, and then what happens is this myromyosin molecule, which has got the ATPase inside, that will break up the ATP into ADP, adenosine diphosphate, and the inorganic phosphate, and energy is released, and that energy is used up by the myromyosin that first makes the contact with the actin filament, and using this energy it will move inward or bend. And this connection between the myromyosin molecule of the myosin and the actin molecule is called as cross bridge. So this cross bridge now will be bending inward. When it bends inward, automatically what happens? The actin filament will be pulled inside, that you see here. The head of the myromyosin attached to the actin at the active site, and now as it has bent inward, the actin filament has been moved inside. And once this happens, again the ATP, another ATP molecule binds to the site of the ATP binding site of the heads of myromyosin. It gets stretched again and comes in contact with the another spot, active site on the actin. By that time, there also, by the help of the calcium ions, the active sites are made open by moving the tropomyosin away, so that further it makes connection and pulls again. So bit by bit the actin filament is being pulled inside. So all the actin filaments which are at the two ends of the myromyosin, now they are moving inside. That is in the space between the myosin molecules. So they will be moving inside like this, and this is in step by step, by ratchet mechanism we call it. These heads of the myromyosin, there are so many heads all along the surface of the myosin molecules. So all these will be making contacts and pulling the actin filaments inward. So that is how the actin filaments from both the sides start moving towards the center of the, what you can say, the H zone. So towards the center of the H zone they will be moving, and ultimately what happens is the two Z lines will be brought very close to the myosin, and that is how, because these Z lines are attached to the actin molecules. So that's why as the actin molecules move inward, the Z lines also move inward, and that is how the sarcomere shortens. And all the sarcomere in a myofiber shorten, the myofiber completely shortens, and like this all the myofibers in a muscle, in a fasciculum, and all the muscles, myofibers in the fasciculi of the muscles contract at a time, right? And that is how the muscle contracts. So this is what the sliding filament theory is all about, which helps us in understanding how the muscle contraction is taking place. Okay, with this we have learned how the muscle contraction takes place. Let us write down steps involved in it. First step will be activation of muscle by transmission of nerve impulse from a motor neuron across the synaptic cleft of neuromuscular junction. That's also called as motor end plate. Once this happens, action potential takes place. Action potential is generated in the muscle fibers, and it is carried to the sarcoplasmic reticuli, which are the storehouses of calcium ions. Okay, and we already learned about this diagram, so let us rub it and write down the further steps involved. Activated sarcoplasmic reticulum, releases calcium ions. Calcium ions bind to the tropomyosin. That is troponin of the tropomyosin. Troponin molecule, and the tropomyosin is pushed away, exposing active sites of actin. Okay, then ATP binds to head of myosin, which stretches and makes contact with filament. Thus, a cross bridge is formed between actin and myosin. The next point. ATP is broken down by ATPase enzyme into ADP, adenosine diphosphate, and inorganic phosphate, releasing energy. Utilizing this energy, the cross bridge bends inward, thereby pulling actin in between myosins. Like a ratchet mechanism, actins are pulled inside. Or actin filaments move inward from both the sides of a sarcomere. The Z lines come to the myosin filaments and thereby sarcomere shortens. Simultaneously, all sarcomeres in a myofiber and all myofibers in a muscle shortens, making the muscle contract. Now this is how the contraction is taking place. The muscle is becoming shorter, and how the relaxation takes place. Once the stimulus or once the stimulation of the muscle cell by the neuron is stopped, then all the calcium ions are withdrawn back into the sarcoplasmic reticulum, and as a result what happens? When there are no calcium ions, again the Locomotion comes back to the original position, and the active sites on the actin are blocked, and the actin filaments go back or move back to the original position. So like that, the actin filaments move away, and again you see that the sarcomeres move back to the normal size, and that's how the muscle becomes relaxed. So this is how the contractions and relaxations in the muscle help us in movement as well as in locomotion.